Hello friends, welcome back to my channel BioFreak. In today's lecture video, we will be talking about ribosomes. Ribosomes are the organelles which are made up of large ribonucleoproteins. What do I mean by ribonucleoproteins is that ribonucleo means they are made up of RNA and they are also made up of proteins. Okay, they have proteins in them. Now what do? They basically translate the genetic information that is present in your mRNA into the polypeptide. Okay, they basically facilitate your translation to occur. Like for example, you have this mRNA from 5' to 3' direction. Ribosome will bind to it and they'll start forming a polypeptide. So basically what they are doing is they're helping you in translation, right? Now they are spherical in structure, okay, roughly spherical in structure, and in E. coli what happens, this diameter of this ribosomes is roughly around 2.5 and in mammalian cell it's 2.8. So as I said, they have this larger subunit and a smaller subunit. It's shown here. This is your larger subunit, this is your smaller subunit, and they have ribosomal RNA, ribosomal protein, it's already said. Now important thing here is that this table, your ribosomes in prokaryotes, okay, they are made up of, overall size of this ribosomes in prokaryotes is 70s, while in eukaryotes it's 80s. And as I said, they are made up of proteins and RNA. Again, it is different, okay, in smaller subunit you have different proteins, in larger subunit you have different number of proteins, different RNA size, okay, in smaller and large. Let's first focus on the prokaryotes. Now in prokaryotes, if you go to add this to 50s, 30s, you will get 80s. But see, what is S? S is sedimentation coefficient. Basically you centrifuge it, and where it gets separated, based on that you find your sedimentation coefficient, which is basically your ratio of velocity and centrifugal acceleration. Okay, now sedimentation coefficient and Svedberg unit, okay, are two different things. Sedimentation coefficient of 1 into 10 raised to minus 13 seconds. Second is the unit of sedimentation. 1 into 10 raised to minus 13 seconds of sedimentation coefficient is 1 Svedberg unit. Okay, now in prokaryotes, as I said, overall size is 70s. In smaller subunit, okay, they have 21 proteins. In larger subunit they have 31 proteins. RNA size in smaller subunit is 16s, while RNA size in larger subunit is 23s and 5s. There are 2 RNA here, and the nucleotide size, okay, the length of the nucleotide of RNA is given in brackets. Okay, similarly in eukaryotes it is 80s. The overall size of the eukaryotic ribosome is 80s, 40s and 60s. Again, if you add it will be 100s. But when these two subunits of ribosomes, which are not always bound to each other, when they bind together, okay, overall size becomes 80s. Okay, they become more compact here and they will get separated at 80s. Okay, now in a smaller subunit you have a smaller subunit individually, if you consider, it is 40s, and larger subunit is 60s. In smaller subunit you will have 33 proteins and 18s RNA will be there, that is 2300 nucleotide long. Here if you see, in larger subunit there are 46 proteins and there are 3 RNA molecules which are there, 28s, 5.6s, 5s. Okay, nucleotides are given in bracket, the length of the nucleotide. Now let's just focus on these 3, okay, 18s, 28s and 5.6s. okay, so 18s, 28s, 5.6s, they are transcribed by single RNA, ribosomal RNA gene. Okay, this is your single transcription unit. This single transcription unit is repeated many times. Okay, there are tandem repeats and there will be non-spacer. This particular portion here is non-spacer, the non-coding spacer DNA. All this RNA genes which are there, no, they are repeated several times and they will form one primary transcript. This primary transcript is 47s, and it has 18s, 5.8s, 28s, okay, altogether in this one particular primary transcript. Later when it matures, what happens? You will get three different RNAs. Okay, three different RNA will be formed like this. One will be 18s, one will be 5.8s, and this one will be 28s. Okay, so like this, when it matures, you will get three different RNAs which you were looking at these three different things. Okay, now where are these rRNA gene clusters located? In humans, if you see in humans, it is 13, 14, 15, 21, 22 chromosome, and these gene clusters are basically present on this nucleolar organizer region, NOR. Okay, so humans contain this gene clusters in NOR, and the chromosome number is 13, 14, 15, 21, 22. Easy to remember. Another important thing is that, okay, this particular transcription of this particular transcription unit is done by whom? It is done by RNA Pol 1. Very important. Okay, it is done by RNA Pol 1. While something remains here, that is 5s. What about 5s? 5s transcription is done by RNA Pol 3. It is present on chromosome 1, the transcription unit for 5s RNA, which is there, ribonucleoprotein, ribonucleic acid. It is present on chromosome 1. So the basic idea here is that you have two different things. Okay, you have 5s on one side and all these three, that is 18s, 28s, 5.6s. okay, this one is transcribed by RNA polymerase 3. This one is transcribed by RNA polymerase 1, present on chromosome 1, while this is present on 15, 13, 14, 15 and 21, 22 chromosome. Okay, so that's the basic difference here. Moving ahead, this is just a table. Okay, so the ribosome source. Now, for example, ribosomes in bacteria, these are of 70s type, chloroplast 70s type. This is the rRNA with the sedimentation coefficient. You will find the rRNA size. Mitochondrial RNA is 55s, while archaebacteria is 70s, like your bacteria. In eukaryotes, we have already seen. So this is the basic thing that we need to understand. Moving further, your eukaryotes, okay, they have two types of ribosomes. Cytosolic ribosomes, which are there, not bound. Free ribosomes are of 80s type, while organellar ribosomes are of 70s type. Organellar as in basically the ribosomes which are present on rough ER, okay, rough endoplasmic reticulum, they'll be 70s type. They resemble the bacterial ribosome, okay, 70s ribosomes which are there. Spatial separation between the ribosomes is also present, like some are membrane bound, some are free. Ribosomes are their functional structural and functional thing is same. Okay, basically they are structurally and functionally identical, but they differ only in the protein that they are making. Okay, where they will be transporting that protein transportation, protein translocation is a big thing. Okay, where these ribosomes are translocating, based on that you will have their spatial separation. Another important term that we will come across is polyribosome. Polyribosome or polysome is basically you have single mRNA, as you can see here. This is a single mRNA from 5' to 3' direction. But the single mRNA has many ribosomes that are translocating in 5' to 3' direction. Okay, they are basically making separate polypeptides, but of the identical polypeptide. Ultimately they want to form this particular polypeptide. Okay, but at a single point of time multiple ribosomes are working in the same direction. They are forming this particular polypeptide. Okay, so that's the thing. Now moving further, okay, the location of the ribosome is important here. there are membrane free ribosomes which are not bound to any membrane. Okay, these are free ribosomes, and they basically synthesize the soluble proteins, the proteins which are soluble. Okay, cytosolic soluble proteins. Extrinsic membrane proteins localized to the cytosolic surfaces, like for example you have your cell like this, cytosolic surface, that means here somewhere if they want to translocate it, extrinsic membrane proteins like actin and spectin, they will be formed by your free ribosomes. Mitochondrial and chloroplast proteins encoded by DNA, they will be basically formed by this particular ribosomes. Peroxisomal proteins are also synthesized by these ribosomes. And next class here, the location of the ribosome is ER, membrane bound ribosome. So the ribosomes which are present on the rough endoplasmic reticulum, they form the secretory proteins. This is important. Okay, when we look into the translocation of the proteins, this is very important concept. Okay, and the integral plasma membrane proteins or lysosomal proteins, Golgi complex proteins. Okay, these complexes which are formed, the translocation of proteins from ER to Golgi. Okay, there are certain proteins also involved in this particular function of translocation itself, that is done by this. Then extrinsic membrane proteins localized on non-cytoplasmic surfaces. Okay, this was cytosolic surfaces, this is non-cytosolic surfaces. So you can see that where that particular protein has to be is to be translocated. Based on that, you have the spatial separation of these ribosomes. And the membrane free ribosomes, okay, they will basically are involved in post translational translocation. This one is proteins found on co-translational translocation. Like for example, what do I mean by this? Okay, we will look into this later as well. It's very important concept like that. So co-translation, as in, let's say you have your mRNA, you have your mitochondria, this one mRNA, and you have your ribosome which is there. It is basically forming this polypeptide. Okay, it is under process. Okay, as translocation starts, okay, as it occurs, at the same time it will start its journey of export. Okay, this particular machinery will start exporting this protein where it was always supposed to go. Okay, it will start entering certain it will start moving out of the cell so that it reaches its location, like that. But this is co-translation. Your translation is in process. Okay, your translation is still in process. At the same time, it has started translocating, it has started moving. Okay, it has started releasing that, and the journey of that particular protein has already begun. Post translation means what? Okay, you have your mRNA, ribosome, you have your polypeptide already done. Both the subunits will dissociate after the polypeptide here is formed. This polypeptide is ready, mature polypeptide with all those signal molecules here, and it will later start translocation. So basically that's the difference. Okay, this is now post translation. So translation process is already over. Now you will start translocating that particular protein. So that was it, and I hope you find this video helpful. Thank you. See you again in the next video.